Dispersal Working Group. He's excited to share the advances made through the collaboration and support by us at ESIIL. So Cameron, welcome. We're really excited to have you kick things off for us.
Woo!
Yeah.
All right.
And we'll be looking up the chat and stuff, but folks can add questions for Cameron in there as we go along too.
Sounds good. You always got to like when you get a woohoo from the audience from over Zoom. So that's awesome. Give me 2 seconds. There. Hopefully you can all see Morel that's shoving spores all over the place right now.
Yeah.
Beautiful. Okay. Thank you, Rachel, and thanks everybody. So I won't reiterate Rachel too much, but I'm here representing our Fungal Dispersal Working Group. And the title of our working group and the title of kind of the collection of work that I'm going to be talking about today is called Advancing Fungal Dispersal Ecology Through Traits and Data Harmonization. And so this is the first time that I kind of took all this work and put it in a one big pile. And what I'm going to try to do is not dive too much in details of the science along the way, but kind of give you and highlight an overview of all the different projects and ways that we took our working group during our time there and where we're kind of going next. Before I dive in too deeply, though, I want to give a quick dedication to one of our working group members, Adriana Romero Oliveras. She unfortunately and unexpectedly passed this past April. She's an integral part of the group and she's an integral part of this work. As the slide says, she's a scientist, collaborator, mentor, but we already think of her as our friend and we miss her deeply within the group, and it was a very unexpected surprise. So anything now coming out of this group is very much going to have Adriana's spirit within it, and we're going to hope to live her on her memory through the work that we do and the science that she contributed to. Okay. This working group was incredible. I've been part of a few different working groups over my career so far, and this group really put the fun in fungal. As you can see in the lower right picture, we really were and developed into a family over the 2 in-person meetings, and we also had a virtual meeting and virtual meetings and coffee hours in between our in-person meetings. We're going to have another virtual conference symposium this coming June, at the beginning of June, to problem solve and think about how we're going to wrap out projects and where we're going to take things next. And our overall mission within the group, as Bala, who is there right in the middle, I don't know if you can see my cursor, and Kabir squatting down, bald in the front row. They were the co-PIs on this work, but it was really Bala driving the working group and then pulled this group all together. And when she approached me and Kabir to be co-PIs, I've worked with Bala a few times over the years. I really love Bala and her approach to things. And her one stipulation for being part of this working group is that there's no a-holes allowed. And so we definitely met that standard. I didn't know many of these individuals as we start out, but now I'd call them all close friends, and we have a WhatsApp group text that we use to talk about science and talk about life and all those good things. Okay. And so the question that you folks might be asking is, well, why are we looking at fungal dispersal, and why do we come to ESIIL look at fungal dispersal in detail? And so what I'll try to impart on you, although I don't know if I need to this group, is that, well, fungi are really important. We have about 3 to 13 million species, and when we think about 3 to 13 million species, only about 6,000% form, or 6,000 form mushrooms and the structures that people traditionally think about of fungi. And so when we think about those millions of species, they serve all these fundamental roles within ecosystems and within our lives. They're decomposers, they're mutualists, they're symbionts, they're pathogens, and they live in a whole variety of spaces. They're terrestrial, they're aquatic, they're marine. And when we think about dispersal, and the cue of dispersal that we were thinking about within this group is that it has broad range applications as well. By understanding dispersal knowledge more deeper, well, we can do things like manage ecosystems, but also think about things like human health. Pathogenicity is growing and increasing in concern, and we recognize fungal pathogens within that realm as well. But a limitation is that we don't have a lot of fungal dispersal knowledge. I'll draw your attention to the middle, I guess, rectangular thing that I made for you called fungal dispersal theory, is that what's happened within fungal dispersal it's been dominated by this microbial approach to dispersal from Bass Becking, where everything's everywhere but the environment selects. And I'd argue working in ecology and teaching within ecology is that that tends to be the approach with dispersal in general. And the way to kind of view that quote is that dispersal is such a complex thing that it's very difficult to incorporate within models and very difficult to understand its contribution to diversity and functioning and all those different things. And so what's happened over many, many years is that we've thrown this kind of shroud over dispersal and kind of created it as this stochastic, unpredictable black box thing. But what we know as we've been accumulating more data over the years on the left-hand side of your screen is that we know that dispersal can add meaningful contributions to things like biogeography and community assembly. Okay. Filter is the first dispersal, or the filtered, my words. Dispersal is the first filter that ecological organisms go through in order to assemble within local communities. So the biodiversity that you see in front of you, the first kind of hurdle that the organisms had to overcome was reaching that location. So it's fundamental in terms of influencing what biodiversity you see in the environment around you. And then as we think about things like environmental change on the right-hand side there, well, dispersal is going to have an effect on that as well. What we're seeing is we're seeing climates and environments rapidly change in faster rates than they did previously. And when we think about previous adaptations to those environmental changes that weren't driven by anthropogenic forces, well, dispersal could probably keep up and evolution could keep up with those changes. But as changes are increasingly rapidly, there's an opportunity there for thinking about assisted dispersal and how we can fit dispersal into some of our management plans. And so when we look at other groups, and sometimes fungi, we have fungal ecologists, mycologists, we have like an inferiority complex because we look at animal groups and we look at plant groups and we look at dispersal and we look at traits and we see that many of them are well integrated into that data. And so when we look at kind of these global approaches, we kind of see these common things arise within dispersal in terms of, well, body size, propagule size, what's being dispersed, what's the size of it, how many, how long does it take to develop, what are the environmental tolerances, and all these different things. So when we look at the animal and plant dispersal world is that we can see these emerging trends and emerging themes, and we've looked at as a group of different ways we can slot fungi into this framework. And when we think about movement ecology is that we're not starting from ground zero within fungi. The way that Bala and I initially got connected is that I did a PhD chapter within my PhD on dispersal, and it was a funny chapter because what we did is we aerial sampled air around ecoregions of biomes around North America, and we found very little evidence of dispersal of arbuscular mycorrhizal fungi, a subterranean fungal group that I work with. And then maybe a decade later, Bala came and she said, I think there's something in the air. And looped me in with a project that she was working on out of Chicago, looking at aerial dispersal within urban systems. And sure enough, what we found within that study is that there's actually prominent aerial dispersal of these groups. And so what's happening is that we're kind of moving into this realm of trying to look deeper into mechanisms of dispersal, tracking within the environment, and all those different types of things. But a rate limiting step is that we need traits. A thing that we've identified within the group is that we kind of went through this decade and a half of sequencing the world. We got really excited because we worked with largely invisible microbial fungi. And so what we've done the world over is we've gone and we've collected soil, water, marine, whatever, air, dust samples, and we've sequenced who's there. But a piece that we're missing, especially when we want to think meaningfully about dispersal, is, well, what kind of traits do these lists of species that we are generating have? And so this is previous work that many within the group have been involved with as well, is thinking about what kind of traits exist within fungi and how do those traits kind of translate to functioning and within assembly of biodiversity, interaction with hosts, or even just general ecological interactions. And so what we did as a group is we did some goals and we said we'd do a few different things within the proposal. And kind of the big themes that we were looking at as well. Goal one is we wanted to synthesize and look at and create data. And we wanted to look at fungal traits and think about how we could maybe organize and create fungal trait database that would be utilized by other researchers and individuals. And then the other goal was to link these dispersal traits to global DNA databases. Again, we have all of these millions and millions of genetic sequences of fungi all over the world. We have data associated with them in terms of geographically where they were sampled, the time that they were sampled, all this metadata associated with them. And so what we were hoping to do is take that one piece of this global data set of all this understanding of who's there, when were they there, where are they, all this other metadata, and link it up with traits and utilize those two kind of big pieces of data to come together to think about how we could meaningfully integrate traits into dispersal ecology and better model fungal ecology. And so the third goal that we had was, well, can we utilize this data and utilize this approach to start making predictive changes? Again, the world is changing rapidly environmentally. And so if we have a better understanding of traits, if we have a better understanding of dispersal, can we utilize all those things to maybe make better predictions of models of how ecosystems and how organisms are going to react to these environmental changes? And maybe down the line, can we apply this in knowledge and information to the applied aspect of managing for things like restoration and conservation? Okay, so that's what we said we would do. And what we actually did is we did a few different things along the way. We generated a lot of questions, and I always tell my students at USC that you're not doing good science unless you're not generating a lot of questions, and we generated a lot of questions, so we did good science, in my opinion, on that end. We developed a conceptual framework and walked through in terms of what that conceptual framework is and where it fits within the larger scheme of the project. We looked at how we could utilize new technologies like large language models to do things like data extraction of fungal traits. And then we put it all together within a predictive data synthesis where we could take our conceptual models, take our data extraction of our traits, take our large global data sets, and can we actually do something with all of that data? So I'm going to start with the questions because that's really where the group started off. What happened was we got together there in that exact room. I have like fond memories seeing it over Zoom right now. But one of the first things that happened within our meeting one within October of 2024 was we had a lot of debates about, well, what questions are important in fungal dispersal ecology? And so what that resulted in was our group, and I'll give a nod of the cap to our group at the end, is that our group's pretty diverse. We spread and range across working career stages. We spread across different expertise in terms of the fungal groups we look at, the aspect that we look at fungal ecology. And so what we decided was that within our working group alone, we have a collection of expertise, we have a diversity of expertise, and this is a pretty good group to survey and get an idea of what are the most important questions within fungal dispersal ecology. And so that was one of the big exercises that we did within our first meeting within October of 2024, is that we essentially, I call it just throwing shit at the wall and seeing what sticks. So we're just throwing questions at the wall, and we generated this big long list of questions that we all thought individually were really important for dispersal ecology. And kind of the underlying theme of the questions that you were developing are, is this a question that is going to address a major knowledge gap and a future research direction within fungal dispersal ecology? And so what happened with that list of questions that we did is that they all got put into a survey, and that's what you're looking at on the upper right-hand corner. Oops, to go back one, there we go. That's what you're looking at in the upper right-hand corner. It was a Google form where some researchers within the group put together a survey to essentially, now that we've made our big bin of 100 plus questions, which ones as a group and I guess, piece of the larger research community, which ones are the 100 most important ones. And so we literally had to rank all of our questions in terms of, well, a 5 is that this is a revolutionary question. This is totally going to like blow the field wide open, and this is going to cook people's noodles and get people really excited, all the way to number 1. It's not a priority. This really doesn't get me excited. It doesn't blow my hair back whatsoever. And so from that list of initial pool of questions that we generated, we finally pared it down to 100 questions based on this survey within the group. And then what happened is that the subgroup that was driving this paper and was led by Sarah Kuprovich, who you can see there, relaxing at the Celestial Tea Factory. She was super excited for the tour because is it Sleepy Time? Sleepy Time was her favorite Celestial Tea, and so she was stoked on that chair. But what this group did is they essentially took all of our 100 questions and then they binned them into these 10 major themes. And so within these 10 major themes, and the paper is published within ISME earlier this year, and you're welcome to go check it out. But within these 10 major themes, what we did is we essentially took our questions and saw where they fit. And so an interesting piece that came out of this is that when we were developing the questions originally, and then when we were going through the surveys and the results of the surveys, is that one thing we noticed is that we don't agree. And that's good. Because again, we have a diverse community, we have diverse expertise, and it's really good that we don't agree on what the most important questions are, because that's how we kind of push science forward in terms of what is going to move and bring it a little bit further along. And so even though we're a little bit fragmented in terms of what we viewed were the most important questions, I'd still argue that everybody's most important question got on the board. It just might not have been the most important question to the entire group. And another thing we noticed is that as we were going through the questions that are emerging and kind of spotting out themes and spotting out patterns, well, we noticed there's a strong taxonomic bias in terms of what fungal questions we were asking, and a strong environmental bias as well, is that we were really limited in looking at things like aquatic or marine fungi, just because it's a new kind of emerging field that doesn't have a big group of researchers yet. And we didn't have one on our team. And so maybe that's a gap that we could address within the future, is incorporate more of these marine and aquatic mycologists within groups. And another kind of themes within the questions is that there's a lack of data harmonization, perfect, lack of data harmonization within the field. And this will come out within this presentation in a little bit, is that there's all this data and information that exists about fungi, but there's all in these like disparate different locations. And so when we put the proposal together, that was one kind of gap that we thought we might be able to address, is being able to harness all of this information and put it in one kind of concrete permanent home. And so, again, we developed all these questions that have got us really excited about, well, what might be next? And so with those questions, what we really recognized too, as we were developing on one of the days within our working group in 2024, is that in addition to like this misconsensus and all these gaps that we're noticing and trends that we're noticing, is that we're really missing really like foundational conceptual framework for connecting all these fungal traits to their actual movement and the movement ecology. And so that started another subgroup within our larger group where they started working within the conceptual framework of fungal movement ecology. And so this work was being led by the PI of the project, Bala Chaudhry out of Dartmouth. And she got this group going because she's been hanging out with a lot of dispersal ecologists. And what she found with dispersal ecologists that work with other taxonomic groups is that they continually come back to this Nathan's Unifying Organismal Movement Research Framework that you can see on the right-hand side here. It was published in PNAS in 2008. And the way to think about this framework is essentially it's all these different components related to a focal individual in yellow. So that yellow circle represents an individual, and that individual has all these different traits related to their movement. There is an internal state shown by the orange block, and within the internal state, those are things that are going to dictate why it would actually move in the first place. There is a blue state, which is the motion capacity. Essentially, how does this organism actually move when that why to move motivation has been reached? The red state is the navigation capacity in terms of like how does it know where to move, or where does it actually move to throughout the environment and what dictates that. And then the green state represents that actual path of where it's moving throughout the environment. And so these are all these internal things that exist for an individual when we're thinking about their movement. But we also have to link it to external things too, which is that lighter blue circle in the top left-hand corner of that diagram. And with that lighter blue, what we're trying to think about is, well, what are the external things that can influence those internal things for the individual? And so this one was a fun exercise to do as a group as well. It started off with, I'm pretty proud of this. This is my sketch here. Me sketching as Ball is talking about this Nathan framework and just trying to think about, whoa, how do we fit fungi into this larger framework? And then it really turned into like this exercise where we were taking all these sticky notes and thinking about, well, what are all these different kind of concepts that we can fit within this internal model? And we literally filled all the whiteboards within the room around you with all these different sticky notes of where our different fungal traits and components might fit within that movement ecology model. And so what it has been harnessed into since then is our conceptual paper, where within our conceptual paper, we're thinking about all these different fungal traits. And so we're thinking about things related to the sporocarp for fungi that produce mushrooms. So for those that are uninitiated, when you hear the word sporocarp, just think of a mushroom. And when we think of sporocarp, there's different traits that could exist within a sporocarp that could affect dispersal. It could be height. It could be what is the cue to release spores from the environment. It could be the smell as an attractant for other organisms, animals to move the spores for you. There's traits related to the actual spores. One thing that's universal about fungi, whether or not they produce mushrooms, is that they produce spores. And we can think about, well, how many spores do they produce? What are the size of the spores? What are the shapes? What are the wall thickness? There's other things like colors that could be integrated as well. And then we also think about things too, like mycelia, because people often default and think about spores as the dispersal mechanism for fungi. But don't forget that they predominantly live as mycelia throughout their life cycle. And so mycelia are also a dispersal mechanism because through those mycelial fragments, new individuals can be developed and produced in new locations. And so we think about mycelial traits as well. And so what we did is we took that Nathan's framework and we thought about, well, okay, with this general traits scheme of different things that can contribute to fungal dispersal, how can we place them within this Nathan framework conceptually? And so it could look something like this, where what we did is we took Nathan's framework and we thought about, well, how can it apply to fungi? And when we thought about fungal dispersal, we thought about essentially 4 key steps that are important for dispersal of a fungal individual. Liberation, essentially what is going to cause the individual to become liberated from their location and freed out to start moving throughout the environment. Transport, so how are they moved throughout the environment? Deposition, how does that dispersal kernel land within the environment in a new location? And then establishment, not only does it have to land there, but it actually has to establish and develop within the new geographical location that it's there where it is. And so here's an example thinking about mushrooms specifically, is that when we look at mushrooms, well, there's many different traits across these different stages that can inform things like liberation, transport, movement, navigation, and motion. So you can think about things like, well, height are really important at the beginning, perhaps the size, the production of spores. These are all things that could really contribute to liberation within a specific location. And then as we go into transport, our second stage, well, now the sporocarp traits are not as important. We don't really care about height, or we don't care about release from that sporocarp because the spores are already out there. So now the spore traits become more important for that transport and then that deposition. And then we get to establishment. Well, the establishment piece that's really important for fungi is mycelial production. Is that it's not only important that that spore lands there, but can that spore germinate and start developing new hyphae, new associations, new ecological interactions wherever it lands. And so we essentially developed this larger framework to think about traits conceptually and think about how fungi can fit within movement ecology. And the goal within this conceptual paper that we're currently wrapping up and finding a home for is to again get folks thinking about a broader degree of traits and how we can apply those traits to a specific ecological mechanism like dispersal. Okay. And so now with our 100 questions and with our conceptual framework, well, we recognize that well, we were brought there to work with data. We better appease the ESIIL folks and work with some data. And so that's what we did, is that we thought, okay, we've developed all these questions, we've developed all these concepts. How can we actually take these things and have rubber meet the road? And so our first limiting step that, again, we identified before we even started the working group is, well, we need trait data, and we need trait data that's going to be scalable, that we can use at a large scale. Okay. And so what happened with this is that it developed another subgroup where within this subgroup it was looking at how we can actually extract data from the literature that currently exists using large language models. And again, I'll reiterate, there is a lot of fungal descriptions that exist out there. And it exists in these really disparate locations. Some are trapped within books, some like this description from a website called MycoWeb of California fungi. They all exist in different locations. And everywhere you look for this data, it looks very similar to this yellow printout that I'm not sure you can all see within the room in front of you. But I think the appreciation you can get from just seeing it within the room, there's a lot of words there. And really, when we look at description of fungi and descriptions of the traits associated with fungi, this is a pretty good representation of what exists out there within the literature. And so you can imagine that it is really difficult when you think about those millions of fungal taxa, but even if you think of those 6,000 fungi that actually produce fruiting bodies, to be able to take all that data, organize it, and synthesize it. I always use the example of like undergraduate labor within my classes, because the undergrads are always keen and they want to do research experience as soon as they're freshmen. I'm like, man, one of your first jobs is going to be probably like pipetting, cleaning, or reading through a description of a species and pulling out trait data. And so what we're trying to think about is, how can we save an undergraduate from having to do that hard labor, combing through all these texts and pulling out important trait data, utilizing new technology. And so with large language models, as they're growing and as they're developing, I don't think I need to tell anybody in the room what a large language model is, but one thing that I will point out about large language models is that they're prone to errors. And another thing with the private ones that exist out there is that they're a bit of like a black box. You put your information within the large language model, you hope that it's doing the right thing behind the scenes, and then it spits something back out at you. And so for something, if you're just trying to write, I don't know, a quick email or something like that, it's okay. But if we're really serious about collecting meaningful trait data, well, we need to be a little more serious about what types of large language models we're going to use. And so in comes your very own Roberto Ramos. He was charging this one really hard, is that he was looking at essentially open source models that you could run locally. Because with these open source models run locally, is that you are the one in control. And he was able to develop this pipeline over time to enable these large language models to operate programmatically, and enable reproducibility, which is really important for science, really important for if we're going to go out and start publishing this stuff and encouraging others to utilize similar technologies. And so what Rob did is he took a database of traits that we already have that's been curated by others within the group. It's called Trait AM. And what it is, it's a global spore trait database for arbuscular mycorrhizal fungi, a subgroup of fungi that exists that many of us work on within the working group. And this Trait AM database, it is one that we would call an expertly curated database. Essentially, this database, we comb through, or the individuals comb through hundreds and hundreds of different texts, and they identified key traits for different species of arbuscular mycorrhizal fungi. And that's the table that you're looking at on the bottom right-hand side. It's an example of what traits those individuals manually pulled out from the literature to build this database. And so what the LLM groups was really interested and curious about was, well, how does potentially utilizing a large language model compared to what we deem as like the expert trait extraction that we have within Trait AM? And so what Rob did is he developed a pipeline to feed in all of the scientific publications that were utilized to build Trait AM. So there's a record of all the different PDFs and citations of all the papers that contributed to the traits within TradeAM. And so what Rob was able to do is he's able to take all of those papers and feed them into a large language model to see how they did. And so he did that. And what he found was what you're looking at on the right-hand side is a series of different traits. And so the way to look at it is on the x-axis, we have a large language model value. And on the y-axis, we have an expert value. Okay. And so the black line that's linear that you can see within each of the graphs, it represents what we expect a 1-to-1 ratio to be. So if the large language model was matching the expert fungal traits exactly, you would expect them to fall right on that line. And he worked with a few different models, and we found was that, well, different models were variable. And overarching across the different models is that it's easier to extract things like quantitative traits using these large language models. But when we think about things like qualitative traits, it becomes a lot more difficult. And so what he's been doing since developing this initial model and this initial data is that there's a preprint available within bioRxiv if you want to check it out. But what he's doing now is kind of massaging it and looking at how we can modify those prompts and modify the large language model that he created to better extract and better represent that trait data. But we were really excited about this because it really opened up an avenue for now we know all this trait data is out there. And now we have a methodology to potentially grab all this trait data and implement it into models. And so that's what the last group is working on, the data group. Within this data group, we're thinking about, well, okay, how can we grab all this trait data and how can we synthesize it and utilize it meaningfully? And within the data group, it's being led by Carlos Trigueros Aguilera in the top right-hand corner there. And this actually happened in our last meeting, in-person meeting. Carlos and I were sitting next to each other one day. And I teach ecology here at USC. And one of the things I teach within ecology, I run a plant pollinator lab where I have students thinking about coevolution between plants and their pollinators within the environment. And so what exists within plant pollination world are these things that we call trait syndromes or pollination syndromes, where if you look at different traits of the plant, so color, nectar production, length of the corolla tube, petal symmetry and diameter, all these things are different traits that we know about flowers. And we know that these different traits have different importance for different pollinators. So for example, if you are producing a lot of color or you're likely trying to attract something that can visually see that color. If you're producing a lot of odor, a lot of nectar, you're probably trying to attract something that shows overnight. So to me, this is an interesting framework because it really shows how you can describe traits of an organism and try to apply and think about those traits in terms of an ecological context. And so what we were curious about was, can we potentially develop a trait syndrome for fungi, but not for pollination, obviously, but for dispersal? And so what we did is we went to MycoWeb, and MycoWeb is essentially an online repository of descriptions of fungi within California, and utilizing a large language model similar to the one that Rob built. This was predated Rob's large language model that he built, but we're going to utilizing it now and applying it to MycoWeb as well, is that we essentially went through and we extracted all this trait data. And that's what you're looking at here, is that on the first column, you're looking at the species identification as it appears within MycoWeb. You're looking at sporocarp morphology, so does it make a mushroom, does it make a cup, a puffball, and then different trait descriptions associated with that species. So essentially what we did is we fed in MycoWeb's information into a large language model, and we told the large language model, please pull out, well, I always say please because I want a good like alternative reality when machines take over. Please, can you extract all this trait data and harmonize it in a workable table so we can apply it to a model? And so what Carlos and I schemed last time we were there is that, well, can we take all of this trait data and can we identify syndromes, essentially groups of taxa that share some common traits within the group? And so first stab at it, what we did is we just generated a PCOA where we essentially identified clusters within MicroWeb where based on the clusters, we could actually separate out species that share traits. And so what you're looking at here is an ordination of what we're designating as our trait syndromes or dispersal trait syndromes, where each color represents a grouping of species that share similar traits. And so we kind of group them within the similar syndrome. And so we identified essentially 6 distinct dispersal trait syndromes just for microweb within California alone. And within these trait syndromes, we have many quantifiable traits, and that's really great from a modeling perspective because what we can do is we can look at these numerical and quantitative traits and apply them to different models. So thinking about things like, well, spore width, spore length, fruiting body height, fruiting body breadth, et cetera, et cetera. And we see that different quantitative traits are more important in some groups than others and more prevalent in some groups than others. But we also identified qualitative traits as well. Essentially, all these weird words that if you're not a mycologist, you have no idea what they mean. Things like teeth, smooth, poroid, parethical, gill, pedicle, truncate, essentially verbal descriptions of traits that exist for fungi. Well, we identified a way to actually quantitate, yeah, sure, why not? Quantitate these categorical traits from the database as well, and figure out how important they are within the syndromes. And so the reason that we chose California is not because I'm from here or live here or work here, but we chose it because one, MicroWeb is a pretty good repository of all this trait data. But two, we have a really good sampling history of ecosystems within California as well. And so what you're looking at here in the top left-hand corner, all those little black dots on the map of California, those are all sampling sites where we have sampled fungal communities within the environment at some point in time, and it's present within GBIF. And then all the other maps that you're looking at are different environmental factors that we also are aware of within California based on historical sampling of the area. So things that are important to fungi like soil nitrogen, organic matter, sand, soil pH, carbon exchange capacity, mean annual temperature, annual precipitation. So we have all this presence data associated with these fungi. We have all this environmental data associated with these geographical locations. We have all this trait data now that we extracted out of MicroWeb. And can we, again, put all these things together to meaningfully predict of what these traits are and how they're contributing to dispersal? And so this is work that's ongoing right now, but where it's kind of landing is we're seeing that, well, there's different sites where different syndromes develop and occupy. And so here you're looking at essentially 6 different maps of California, where within the 6 different maps, it's where we're finding our 6 different trait syndromes of fungi. So you can see that if you look at trait syndrome number 3 in the top right corner in blue dots, is that they're not as widely dispersed as trait syndrome 2, which is shown in green. And so what Carlos is doing right now is he's taking all this geographic data, this presence absence data, and this trait data, and he's plugging it into an HMSC model to see if we can utilize these trait syndromes to meaningfully predict of where these fungi will disperse. And eventually utilizing this model and utilizing this data to use it as a predictive tool of how are these communities potentially going to respond to things like environmental change or climate changes or whatever it may be. And so that's what I call, on my drive over, I came up with this one, our 4 pillars of fungal dispersal. The way that we kind of developed it along the way is that we were really excited to jump into data, but what we recognized is we had to walk before we ran, and that's why we started with 100 questions, then conceptual frameworks. But really, those have acted as the anchoring to inform the boots on the ground work that we're doing now, looking at how we can purposefully and meaningfully extract all this data and then utilize it to predict occurrences of fungi, but also build meaningful models of what we call purposeful dispersal. And with that, I'll have to, again, give a nod of my cap to this amazing group that I'm a part of. Like the quote says, if you want to go fast, go alone. If you want to go far, go together. And that's definitely what this group does, is that we go together. And again, we're meeting again this June to try to wrap things up and think about what's next for the group in terms of where we're going to take this work and where do we see the field going after this. Thanks so much.
Keep an eye on the chat too.
I love your syndrome idea. Super cool.
Thanks, Ty.
Help me understand ecologically what a pollinator is to these fungus. What are the things that the syndromes are selecting around?
So one way, that's actually a good, keeping that pollinator piece in mind. When you think of a pollinator, what do you think of, Ty?
Individual with preferences that physically move stuff.
Yeah, it's another. Perfect. It's another individual that's moving and doing something for the plant, right? So a way to think about and try to make that connection potentially with pollinator syndrome is that there's some fungi that, well, they're subterranean and they really don't rely on above ground liberation of spores. They really rely on animal dispersal. And so what we recognize is that, well, there's trait syndromes that exist for those subterranean fungi where they have traits that make them more likely to be dispersed in that mechanism, dug up by an animal and moved in some different location. Versus with others, well, the dispersal syndrome that they have is more for that long distance aerial dispersal. And so you can think about things, well, like a really large sporocarp that's high off the ground that produces these little tiny spores that can be liberated and moved at further distances. And so there's all these like little subtle traits that exist between the different trait syndromes. And so what's be interesting is that we're going to get some of the more hardcore mycologists within the group. I call myself a fake mycologist. I'm more of an ecologist. But we're going to get some of like the hardcore mycologists looking at these trait syndromes and thinking about ecologically, how can we potentially like designate these different syndromes?
Cool.
I'm not sure if that answered your question or...
Oh, that was awesome. Love it. Thank you.
Cool.
I'll ask one. This is really cool, thanks. So like the slide that you kind of ended on with the 4 pillars seems very compelling and seems very like, wow, this all just like worked out so well. I'm just curious how much of your time span was like spent just putting sticky notes all over the wall and figuring out what the hell you're doing and all that kind of stuff. Like how much time did you all have to spend doing that kind of really just like searching for what the purpose is going to be ultimately of the working group compared to like where you ended up now? Is it a pretty even split, or?
No, actually, it was front loaded, all that creative work and sticky note work, because that really was only 2 days. 2 days within our first meeting in October 2024, where we decided within these 2 days we are intensively going to throw darts at the board and see what sticks. And so after we did that, what we decided is that everybody contributed to every one of these projects, and so everyone kind of like initiated these subgroups at the onset, and then what we did is we subdivided the labor to conquer the different aspects of it. And so what happened was with the 100 questions and the conceptual paper subgroups is that they took all those initial inputs and went running with it in terms of developing the actual 100 questions paper and the conceptual paper as well. And so it was really, again, like 2 days of the group contributing early on to think about those concepts. And then I guess a year and a year a bit plus taking all that information and synthesizing it in a meaningful way in terms of the 100 questions and a conceptual paper. And then the other group, so the data group was really the first one that is originally formed in 2024. But as we were trying to figure out how to meaningfully like get this trait data and start developing a model that we could tell people to utilize and hope people would want to utilize, as we recognized that we need to figure out a way to scalably extract traits. And so that created another subgroup that Rob really took the charge of developing this large language model. And so both the data and the large language models, they take time because, well, it's computing time, it's processing time, but it's also like kind of pushing the boundaries a little bit. So it takes time in terms of thinking about, are we doing the right thing? What is this model showing me? Is this ecologically meaningful, or is it just like an artifact that I created based on whatever? Yeah. So early on, like our scheming was a couple days in that first one, and then we really hit the ground running. And then an unfortunate thing when you all go your separate ways is life takes over and individual work takes over. And so what we decided to do, like do keep yuppie with our work and make sure that we're still pushing the boundaries, is that between our October meetings, we had a June meeting where we met virtually over Zoom over 3 days and kind of checked in and worked intensively on the different projects. So then when we came back together in October 2025, we hit the ground running and we were working hard on the different subgroups to try to get these things to the finish line as best as we could. And then again, we're not to the finish line yet for these different projects. And so we're meeting again this June, so next month, to again have a mini virtual group, working intensive group to push all these things to the end line.
Cool, thanks.
Well, there's a silence in the room. I really want to thank ESIIL as well, and all Rachel and Ty and all the other support folks at ESIIL. I will say that from a participant's perspective, it was really well done and really well put together, and it really enabled an opportunity to do a deep dive into science, which I really love, and really, I think, facilitated the cohesiveness that we see within this group too. So a big thanks to all of you for all of your support and creating this program in the first place. It's been really great for us.
Yeah, thank you.
Any final questions for Cameron while we have it?
You can always reach me on email. They have my email, so by all means, reach out if you have any questions, and I'm happy to chat anytime.
Can I ask one more question? How do you think fungal dispersal is different now that you've had your working group?
Well, it's a lot, and I had this before, but now I feel like there's actual framework to back up this thought, is it's purposeful. And it's not this black box thing. It's something that has, we can predictably develop models that will predict fungal dispersal. Okay, it's not the stochastic black box thing. It's a purposeful ecological mechanism that fungi do. We just need the better language and models to be able to describe what it is. And so I think what we've uncovered within this work is that for sure, this thought that Bala and I had exists in terms of there's got to be meaningfully, ecologically predictable ways to describe dispersal for fungi. And so I think we're pushing and edging towards there. And that's what we're really hoping to do some ground truthing with our data paper and our modeling work.
That feels like a pretty fundamental change in the way people are thinking about, I mean, the discussion really has always been fungus is everywhere and it's selected by the environment. So you're really pushing back on the historical paradigm, right?
Yeah, it's, and again, I don't think, you talk probably to most of my ecologists and they'd agree that dispersal is purposeful, but again, they don't have the framework to describe how it's purposeful, if you know what I mean. So I think if you talk to any of my ecologists today, they dump on the Bass-Becking hypothesis, is that, well, ecologically dispersal is a meaningful thing in terms of what the organism does, but we can't describe what that is. And so our goal within this group is, well, we need to give people the script and the understanding of what actually happens to influence that dispersal.
Sweet. And last thing, would you have any recommendation to future working groups or existing working groups on how to be successful?
No a-holes is really important, and it worked out really well. I think, well, it's just like anything, any relationship, it's open communication. And I think one thing that was really useful is early on in our first meeting, we set a lot of ground rules and we spent a lot of time thinking purposely before we even jumped into any science of how we were going to be as a group. And so some of that was utilizing the framework that you folks provided through ESIIL, but some of it was also provided through Bala and Sarah within our group have done working groups together before as well, to think about group norms and group interactions and thinking about your role within the group and how you operate. So it was very much like a psychological experiment in terms of thinking about, well, how can we work cohesively? Who are you as an individual and how do you work with others and how can you put those two things together? And so we really, again, we front-ended that. I'd say for our first day, we didn't talk about any science whatsoever. We just talked about how are we going to work as a group and how is this going to work. And then after that, we jumped into the science.
Awesome. Thank you.
Amazing. Thank you, Cameron.
All right. Thanks, everybody. Really appreciate it.